Hi everyone, welcome back. Now that we've gone over transcription and RNA processing, we're going to spend today talking about translation. So just as a review, we've already gone over part of this. We have DNA, right? DNA is found in the nucleus, that's where it's protected, because DNA is the blueprint, and we only have the DNA that's in the nucleus. So to protect it, we make RNA. mRNA is capable of leaving the nucleus. mRNA basically is the copies of DNA. It's the DNA's message. And that's a process called transcription that makes mRNA. mRNA, once it leaves the nucleus and goes into the cytoplasm, is going to produce proteins using a ribosome. A ribosome is a ribosome factory. It's a protein factory. And the mRNA that meets up with the ribosome is going to make our long protein chains that we learned about earlier in the year. That's going to be caused by a process called translation. And of course, proteins are going to make, my head's in the way there, but proteins are going to make all the traits that we have. All right, so moving on. Translation is from nucleic acid language, specifically RNA, to amino acid language. So that's proteins. So we are going from RNA to proteins. What we see right here, I'll use the pen to highlight what we're talking about. We are talking about this part here, from RNA to protein. Okay, so how does mRNA code for proteins? Well, here's our DNA sequence. It starts with TAC and ends all the way on the right with CGG. And DNA is going to produce mRNA, right? So we already know how that happens in transcription. We know that RNA polymerase is going to add RNA bases to a single strand of DNA, a strand that's been pulled apart. So what's in green here, this is going to really be the complementary RNA bases to the DNA you see above. Notice it starts TAC, so the mRNA begins AUG. All right. So RNA, then once it is produced, is going to make proteins. And these proteins are going to be coded for by the mRNA. But the question here that's kind of blocked by all this stuff is how can you code for 20 different amino acids with only 4 nucleotide bases, right? DNA has 4 bases, A and T and C and G. Over here, mRNA also has 4 bases, A and U and C and G, but proteins have 20 different amino acids. So how do we go from the language that is just 4 bases long to a language that's made up of 20 different amino acids? Well, that was a problem that was figured out a while ago when people realized something about mRNA, and that's mRNA codes in triplets. So we have to think about the mRNA bases as groups of 3. So AUG, followed by CGU, followed by GUA. You can separate them with a line. I think that's a good way of showing the difference. Each one of these triplets is going to code for an amino acid. And there we see the proteins, met, arginine, valine, and so on. So it's the triplets that matter. All right, we call a triplet a codon because it's like a coding unit. Okay, so Crick of Watson and Crick fame discovered the triplet codon system. So he discovered that the codons could be read in groups of 3. Nirenberg and Khorana determined the mRNA amino acid match. They figured out which codons coded for which amino acids. So they sort of cracked the code of translation. They added fabricated mRNA to a test tube that had the necessary components. And they created artificial UUUUU mRNA, and they found out that all of the U's coded for phenylalanine. So they did this to figure out what the code was. There they are, the people who cracked the code. And this is how we are going to crack the code. This is the decoding device, the codon chart. So here we go. This is the code for all living things. So this is how we decode mRNA into proteins. And it doesn't matter if we're talking about humans or trees or shrimp or bacteria, mold, or whatever. Every single living thing uses the same code. It's actually evidence for the unity of all living things, right? It's probably not a coincidence that every single living thing uses the exact same code, just like this that you see here. So it's a strong support for the common origin of all life. The code is redundant. So this means that the code can be repeated in some examples, which we'll talk about. 2 or more codons for each amino acid. And this is because of something called a wobble. So let's talk about the wobble before we go over this. So let's look at serine, S-E-R. Serine is what I'm highlighting in red. Serine can be coded for by 3 different codons. What are these codons? The first one is UCU, followed by UCC, and then UCA, and then UCG. Right? Notice what they all have in common, the U and the C, but it's the third letter that's different. That is what we refer to as the wobble. So Francis Crick, who discovered the wobble, realized that maybe it could be it could have happened, and then evolution could have kept it in there because it helps cover a few mistakes. So we see that they don't all have the same amount of wobble. So we can see that, let's see here, alanine also has 4, but something like leucine only has 2 different codons to code for leucine. Methionine only has 1. Methionine is the start codon. It's the very first one. Tryptophan also has only 1. Histine has 2. So depending on the amino acid, there's 1 or 2 or more codons for each amino acid. All right, the start codon is AUG. Every single coding region on mRNA will always begin with AUG. I like to remember that by remembering the school always begins in August. August is the beginning, and it codes for methionine. Methionine is that first amino acid. Then there are stop codons, the stop codons, UGA, UAA, and UAG that you can see there highlighted in white. These are all of the codons that will end an amino acid chain. So this tells the ribosome that the amino acid is done. Right? Now, how do you read this chart? So the first base is on the left. So if you're reading this and your first base, let's say we're looking at GCA. So we first look on the left. So I'll use my laser pointer. On the left is over here. So I want to find G. There's G. So it's somewhere in this row. And then I need to find C. C is up here. C is in this column. So I must be somewhere in this box. GCA is right here. So I must be referring to alanine. So that must be alanine. Okay. So how are the codons matched to amino acids? So what's the actual mechanism that takes a codon and matches it to an amino acid? Well, that is where something called tRNA comes in. tRNA is a specific type of RNA that has a little area that will have a complementary pair to the codon. You see here how the AUG on the tRNA will have a UAC. It matches up perfectly with that first codon. And this tRNA has the correct amino acid, methionine. All right. And then we have the next amino acid, arginine. Again, the tRNA matches up perfectly with the codon. The CGU that codes for arginine happens because the tRNA has these bases that happen to have the exact right pair. Valine, these bases on the tRNA are referred to as the anti-codon. The anticodon is found on tRNA. The codon is what we really care about. That's the actual stuff that we will use the chart for. The codon is found on mRNA. The anticodon is found on tRNA. Okay, so to review, we've got our DNA. We've transcribed it into mRNA, the copies. The mRNA is now being used to make a protein, but we've got some help with a ribosome and with tRNA that's helping to bring it. And this is going to help make our protein, which we haven't quite done yet. Okay, so more about tRNA. tRNA is transfer RNA, and it has a clover leaf structure. There are a few different views here. On the left, you can see the clover leaf view. It looks very clear. Then we have some other views on the right. The most common one you'll see is the one I'm highlighting here. This is like what I call the candlestick view. But either way, you've got an anticodon on the clover leaf end. So that's like the bottom of the tRNA. So the anticodon is down here. So notice this one is AAG. That's what's going to help it match up to the codon. Here's the anticodon. And again, there's the anticodon. So the anticodon on this top picture would be here. And then we have the amino acid attached on the 3' end, which is the opposite end, there and there. It's like the wick of the candlestick. Okay, in addition to tRNA, we also have ribosomes that work to help build these proteins. Ribosomes really hold everything together. Ribosomes facilitate the coupling of tRNA with mRNA. So tRNA and mRNA are able to come together with the help of a ribosome. So is a ribosome an organelle, which we described it as before, or is it an enzyme? Well, it's a little bit of both. It does both. Sometimes a ribosome is referred to as a ribozyme. A ribozyme is an RNA-based enzyme. And in fact, a ribosome is probably a ribozyme because it does have enzyme-like capabilities that's performed by the RNA that makes it up. Ribosomes are mostly RNA. Remember rRNA that we talked about before? rRNA makes up most of the ribosome, but there is protein in there as well. Ribosomes are made up of 2 subunits, a large and a small, and they come together right around the mRNA. See that in the picture. All right, ribosomes have 3 sites for tRNA. So there is the A site. This is the entry site, like the first letter of the alphabet. This is where everything goes in. So this holds tRNA that is just entering the ribosome, and it's carrying an amino acid. So the amino acyl tRNA group site. The P site is the middle one. That's the one you see right here in the picture. This is the peptidyl tRNA site. So this is holding the growing polypeptide chain. There's the P site. And then you have the E site, the exit site. This one is empty because when a tRNA gets to the E site, it leaves, it exits. It's the E site. So when we build a polypeptide, first of all, the very first step, the initiation step, is going to bring together the mRNA, the ribosomes, and the tRNA as well. It's all going to come together. You see that in the picture on the left. So we see the very first tRNA with the ribosome. Notice what's the codon, right? The codon is AUG. That's our start codon, isn't it? So the codon is AUG, then the anticodon has to be UAC. You see that right there? And that's going to have methionine, because methionine is the first amino acid. And then we can see our next tRNA is being brought in to the A site. And there in picture 2, we see that next RNA is there on the A site, and the ribosome is going to move down in picture 3. See how the ribosome has shifted over. And now we are adding amino acids in the correct order. Notice that in picture 4, on the P site, the site with a peptide bond, we see that growing polypeptide chain. This one on the end is about to exit. And this just continues. More tRNA enters the A site, and we make the amino acid chain longer and longer and longer until we get to UAA in picture 5. That's the stop codon. The stop codon will have a release factor go in, and that release factor makes everything fall apart. And that's basically the end of translation. All right, so that is how translation works. After the protein is made, if it's being made in the ER and it's going to go somewhere else, then it needs a signal peptide, so it needs like an address label to tell the cell where to take it. It could be secreted, it could be going somewhere else. Some proteins are going to stay in the cytoplasm, as we know. Some are going to go to a chloroplast or a mitochondria. Some proteins might go to the cell membrane. There's many, many places where we could send a protein. All right, so what is going on? So we've got a few things happening here. I went a little bit too far. Okay, so we have our DNA in the nucleus. The DNA gets read by RNA polymerase. That RNA polymerase is going to make pre-mRNA, which is going to have the introns cut out. It's going to get a cap and a tail. And it's going to become mature mRNA, which can then leave the nucleus. And once it leaves the nucleus, it's going to meet up with the 2 different ribosomal subunits, which are going to come together with our tRNA to make a long polypeptide chain, which is going to make an amino acid chain. So there we see that growing polypeptide. Okay, so here is our, just to give you an idea of everything, here is our gene. So this is, in blue is the DNA. Our gene is highlighted in blue. That's the Tata box, that's the promoter, if you remember from before. The enhancer is way to the left, not on the screen. And RNA polymerase meets up at the promoter. This is where transcription starts, this is where it ends, and the RNA polymerase is going to travel along, adding in the RNA bases, and we are going to end up with mRNA that has to cut out the introns, splice together the exons, and it's going to give us pre-mRNA. Right? So our pre-mRNA is going to turn into mature mRNA. We're going to get a cap and a tail, and that is going to be used at the beginning and at the end to protect it. And then a ribosome is going to meet up with a mature mRNA, and we're going to make a protein from this. So that is the transcriptional unit. Okay, in prokaryotes, things are a little bit different. And that's because prokaryotes don't have a nucleus. So there's a big difference between prokaryotes and eukaryotes. Prokaryotes keep their DNA in the cytoplasm, and as we know, eukaryotes keep their DNA in the nucleus. Prokaryotes have a circular chromosome. Eukaryotes have linear chromosomes, many of them. Prokaryotes have what's called naked DNA. That means no histone proteins attached to their DNA. And eukaryotes have DNA that is wound around histone proteins. Prokaryotes, there's no introns in a prokaryote, but there are introns in eukaryotes. Okay. So the introns have to come out in a eukaryote, but that's not going to happen in a prokaryote. So this actually makes transcription and translation much more simple in a prokaryote because these 2 things can happen simultaneously. The mRNA doesn't have to be spliced, There doesn't have to be any processing. We don't have to worry about histones on the DNA. We can just make the mRNA, and then we can attach the ribosomes right to the mRNA as it's being transcribed, right? There's no exiting the nucleus into the cytoplasm. There's none of that in a prokaryote because there is no nucleus. So these ribosomes are going to translate as that mRNA is being transcribed, and you can see that on the picture below. So the DNA is in the cytoplasm, there's no editing, and the ribosomes can read mRNA as it is being transcribed. All right. So the time and the physical separation between the processes keep RNA and DNA different, and there's no processing. All right. That is all we have for today. So if you have any questions, please let me know.